Okay, we're running.
Yep.
Yeah, okay, welcome everyone. My name is Sibren Otto from the University of Groningen, and on behalf of the Origin Center in the Netherlands, I welcome you all to a lecture by Jim Cleaves. So we're really delighted to have Jim here. So Jim did his PhD in 2001 at the University of California in San Diego. Also, I think associated with NASA already at that time, and then he stayed on as a postdoc with NASA, and then through various other positions, started in 2013 at LC in Tokyo, the Earth Life Science Institute, where he is a specially appointed professor. But at the same time now he also holds an affiliation with the Carnegie Science Institute at the Earth and Planets Laboratory in Washington. So Jim, again, we're very happy to have you here, and I really look forward to what you're going to talk to us about. So the floor is yours.
Thank you very much, Steve. So I'll shoot for about 25 minutes, is it?
Yeah, that's perfect. I think you have half an hour, and then we have half an hour discussion.
Okay.
So by the way, yeah, good you mentioned this. People that want to ask questions, I would prefer if you have the opportunity to also ask them live, but it's also possible to type them in the chat as we go along or after the lecture. So whatever you prefer and you're most comfortable with.
I'm not sure I can see the chat window from my view, so if you see something, just stop me and bring it up. That's fine. All right, so good morning or good afternoon, as the case may be where you are. My name is Jim Cleaves. I'm here to give you a little bit of a view of the kinds of work we're doing on trying to interpret complex abiological organic reactions as they may be relevant to the origins of life or the detection of life on other planets. So the kind of the big picture question is, we have organic material that can be produced abiologically, and we have biological organic material. Biology recapitulates itself, so we understand the source of biological organic material in the context of biology. We don't understand the origin of life, how abiological organic material or abiological inorganic material becomes biology, but we also know that there are a wide variety of environmental processes that can make complex organic compounds. And we know this from measurements of things like the organic compounds in meteorites that fall to Earth that we're able to analyze, examinations of things like the atmosphere of Titan, various in situ measurements that have been made on comets and asteroids and so forth. So we have these two suites of things that can be very complex and diverse in terms of their organic composition. We want to go look for life in other bodies in our solar system and ultimately beyond our solar system. How are we going to tell the difference between a complex set of organic molecules that come from some natural environmental process and those that are derived from living processes, right? And so I think this involves trying to understand a whole bunch of things at once. One is the process by which natural processes can make organic compounds and what kinds they do, which involves understanding the reaction processes that create that kind of transition. Also understanding how those things degrade in the environment. So if there's a pulse of synthesis, how do those things break down? This is, of course, a topic very central to organic geochemistry because it's important in the provenance of things like petroleum and coal, right? So sometime long ago, natural biological material was buried and it has been biogenized and processed by geologic processes. We get very complex organic mixtures, which are not very similar to their source material in many ways. Right? On top of that, we want to understand if we want to build models for the origin of life. It's probably a good idea to understand what the starting materials that are available are, so we can sort of understand how those interact with environmental systems and with each other, how they potentially self-assemble. And all of these bits of understanding can contribute to methods for determining whether, when we do find or return a scoop of soil from Mars, we can say this contains biology and it is extraterrestrial and not contamination, right? So these are all be little pieces of little clues to contextualizing that information when we get it. So in the context of early planetary history, in particular on Earth, we think there are a variety of processes which might have contributed complex organic materials. Things like atmospheric syntheses, depending on the composition or the oxidation state of the atmosphere, different types of energetic input, including lightning or ultraviolet radiation and so on, can convert those gases into more complex molecules and rain them out to the surface. Likewise, we know that there is today a constant input of extraterrestrial organic material in the form of interplanetary dust particles and meteorites. This flux was probably much higher on the early Earth. Understanding the composition of that material involves understanding solar nebula processes. We also now have a rougher understanding of how geothermal and geochemical processes can actually create reduced complex organic compounds via the interactions of reduced upper mantle materials with subducting seawater. Right. But among all those different processes, which may produce relatively different types of organic mixtures, we also then have the kind of the churn of this stuff in superficial environments. We have oceans which may be of different pHs or temperatures, different salt concentrations. We may have localized environments where we have fluctuating conditions. So there's a lot of opportunities for these already complex materials to complexify further in the environment, right? And so I hope you can appreciate just, it's sort of a spider web of potentially interacting processes. So why I present this as a background is our analytical chemical techniques really have a very strong effect on how we perceive what the problem at hand is, right? If we want to construct a model from the origin of life, it seems reasonable to say, well, what were the compounds that were available? And then let's see how we can put those together into some sort of living system or evolvable system. So I like to take as an example the Miller-Urey experiment. If you're not familiar with this, it's kind of iconic chapter 1 in your biology textbook sort of experiment. What Miller did back in the 1950s was he constructed this flask where you could put reduced gases, which were thought to mimic the early atmosphere, into this closed apparatus. You had another bulb that contained a water reservoir. There are 2 electrodes inserted into this apparatus where an electric discharge can be passed. This water gets boiled, simulating the hydrologic cycle on the Earth. So you have steam generated, which condenses in the discharge flask where the gas is. Whatever water-soluble molecules get made there condense into the steam, and those are returned to the simulated ocean reservoir, right? It's a continuous cycle. And what Miller found, using a mixture of ammonia, methane, hydrogen, water, was that he produced a measurable yield of biological amino acids. Using the technology of his time, which was paper chromatography with anhydrous spraying, he was able to detect aspartic acid, glycine, alpha-alanine, traces of beta-alanine, and alpha-amino N-butyric acid. Okay. Not to disparage that at all, it was a feat of chemistry for its time, but we can now go back and look at these same reactions, which you can see are visibly full of goo, and measure them with more sensitive techniques, such as liquid chromatography, mass spectrometry, with fluorescence derivatization. And we detect, in addition to the amino acids that Miller detected then, a whole host of other compounds that we're able to identify with standards, in addition to a whole suite of other things that we can't readily identify. So it's kind of like diving a little bit deeper below the iceberg. But to take that to another level, we can also just take the raw goo from this kind of experiment and inject it directly into a mass spectrometer using electrospray ionization. And we see that, in fact, there are probably tens of thousands of unique mass compounds present in these mixtures, right? Limited by the resolution of the instrument. So here we're seeing it just, this is a 1958 milliliter electric discharge experiment. We're seeing intensity on the y-axis and M to Z ratio, or roughly mass, on the x-axis. And you can appreciate that there's a very fine resolution here. We're seeing compounds at almost every conceivable formula mass, right? And what's important to realize about this too is if you have a good enough mass spectrometer, you really can resolve a mass well enough to get an elemental formula for the species you're detecting. That doesn't mean that that formula doesn't represent multiple isomers. I'll talk about that a little bit more as we go down the road. But I want you to keep that in the back of your mind. So what's interesting about reactions like Miller-Urey, whether you think they are representative of the early atmosphere or not, is that they're examples of diversity generating reactions where there's a system of chemistry that gets set up, wherein you start with a few reactive molecules and those are able to cross-react and then create new reactive molecules, which are then able to cross-react, and create new reactive molecules, which are able to cross-react. And so you get this kind of combinatorial explosion of products. And we see similar sorts of phenomena in carbonaceous meteorites, that they contain a really wide abundance of structurally related compounds or classes of compounds. So we think that they have a similar provenance, that there's some sort of diversity generating reaction that has exploded at some time during their history. So what we want to do is build techniques to be able to deconvolute these really complex mixtures, right? Rather than being able to say, well, I have a standard of glycine, is there glycine in this mix? We want to understand what the tens of thousands of components are, right? So we can say something deeper about the actual composition of these reactions. And so in the context of looking for life using organic biomarkers, we can think about biochemistry as, in a sense, also a diversity generating complex reaction network, right? That through the set of biochemical processes that life encodes, it takes a few simple inputs and it turns them into this wide variety of components that help life make its way. So are there some simple metrics that would allow us to tell the difference between a complex mess made by biology and a complex mess made by non-biological processes? Right? That's sort of the crux of the question. Right. So I'll give you a brief intro to the concept of chemical space and what it is. A chemical space is an idealized set of compounds that are described by some set of attributes, right? So we can say, for example, what are the number of possible drug-like compounds that can be constructed from organic chemistry? And what is a drug-like compound? Well, it's defined by some solubility limit, some ability to pass the blood-brain barrier, some number of hydrogen bond donors and acceptors, which allow it to be a good binder to an enzyme, active site, and so on. Even using all of these cutoffs, it's estimated that the number of possible drug-like organic molecules may be on the order of 10 to the 33 to 10 to the 180. Just to give you some astronomical reference to that, this is much, 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 much larger than the number of stars in the universe, right? So it is in that respect really remarkable that we have any medicines at all, since we've obviously sampled a very small area of the space. Importantly, what our chemical knowledge has described so far, this is a little bit dated now, we're about a decade out of date, But the Chemical Abstract Service described in 2009 about 5 times 10 to the 7 discrete organic and inorganic compounds. About on the order of a few 100,000 natural products are known to science, which is, again, very small compared to the number that's possible. And it's estimated that the Murchison meteorite, a sort of touchstone exemplar of pre-solar organic chemistry, contains maybe 10 to the 5 to 10 to the 7 unique compound types. Right? Importantly, for reference, what's known as the common core of modern metabolism, that's what's shared between bacteria and whales and humans and so on, is only about 500 compounds, right? So it's 20 amino acids, a couple of common cofactors, the ribonucleotides, the deoxyribonucleotides. Not to say that those aren't strung together in very complex ways to give unique macromolecules, but the small molecule building blocks are really quite a small set. Okay. So I'll give you a quick intro to matrix representation of organic chemicals. It turns out to be a happy fact that we can represent organic molecules as graphs, mathematical graphs, in the sense that a molecule is a graph in that the atoms are nodes and the bonds are edges, which allows them to be represented very precisely in computational languages and formats, which allows us to manipulate a lot of chemical exploration in a very precisely and easily automatable way in computers. So, for example, it's very easy to represent a molecule like glycine as a connectivity matrix, where we're going to describe the atom numbering here, which atom it's connected to, and then the multiplicity of the bonding in a very simple matrix, which can also designate which atom types each atom number represents. And so some potential uses of this kind of representation is, we can enumerate all of the matrices that fulfill certain types of atom compositions, as long as they satisfy Lewis electron bonding rules, or we can codify organic reaction transformations as being transformations where you recognize certain motifs in one matrix and certain motifs in another matrix, and recognize that those can be combined together to give product matrices, right? And you can generalize this so that you don't need to encode it for every particular molecule. It's just a recognizable motif. Right? And so this allows you to screen very large libraries of molecules for potential reactive isomers. As an example of the application of this, we can take a finite formula like that for the natural ribosides, describe certain attributes that a potential genetic material building molecule needs to have, and then build all of the isomers using these kind of algorithms. And that allows us to describe the chemical space of informational molecules that are similar to ribosides. Then we can ask questions about what is special or potentially better or worse about the one that biology actually uses. Right? And it can give us leads to construct new kinds of molecules and test how they behave in the laboratory. All right. So back to the issue at hand of trying to understand what's present in these complex mixtures, using these same sorts of generational algorithms, we can compute the entire possible organic isomer space of any given molecular formula. Right? We did it for a particular formula in the last slide. Now we're going to compute every possible structural isomer of molecules that contain certain combinations of atoms. All right. So, for example, we'll just compute the space of molecules that contain carbon, hydrogen, and nitrogen. Here we have carbon, hydrogen, nitrogen, oxygen, and sulfur. I should say dry is we allow certain bonding motifs that we consider to be unstable in water in these computations. Wet in this case is where we consider things that we would not consider to be stable water. So it lowers the isomer space a little bit. Why is this useful? Because if we're going to go look at these complex things like Miller-Urey experiments, even looking at relatively low masses, like 150 AMU, we can see that there's potentially a million-fold redundancy in the isomer space. So even if we can resolve a peak with a high-resolution mass spectrometer, that may not tell us that much about what that peak represents, right? It could be anyone in various ratios, a combination of potentially a million different isomers, right? So, 10,000 peaks by a million isomers tells you you really still don't understand what that is very well. All right, but we had this thought that what if we could use some knowledge of reaction chemistry to cull this space? And that is to say that any given mass can't be any given molecule in a complex mixture because it has to be made by a process. And we know something about organic reactivity. So if we could make a map of how we think a chemical reaction unfolds, we can then ask, is there a component in this map that has that mass? And that now gives you a lead to say, does this mass peak, contain compounds that have that mass from that reaction map? Hope this is making sense. So we built a computational workflow that basically does exactly this. You begin with a set of defined reaction rules. You can define the input reactive molecules. Right? That could be methane, water, blah, blah, blah. And then you allow them to react with each other. You take the products of that initial reaction, you throw them back into the hopper and allow them to react with the initial reactants again, and so on and so on. You iterate this process, and you gradually build up a rather complex reaction network. And then you can go through and evaluate the composition of that network with respect to real-world experimental data. And then having this map also, you can compute the properties of the molecules that are produced using chemoinformatic descriptors. And you can also, now that you have a map that is actually a process map, you can go in and look for things like autocatalytic cycles, and you can evaluate whether they are thermodynamically favorable autocatalytic cycles and so on. The nice thing about this whole map is this workflow is it's open source. We hope to post it on GitHub by the end of this week. We're trying to just dot the I's and cross the T's on the paper. But that's the idea. And you could drop in any set of starting materials and use any reference spectrum to do this kind of evaluation. And at the core of this workflow is the mod software developed by Christoph Flamm, Jacob Anderson, and colleagues at the University of Southern Denmark. And again, how it works is these kind of matrix representations of molecules are used to make generic reaction rules that allow you to anticipate how two molecules will react. And I should say, I'm a very heartwarming outcome of COVID was that I had a very large number of virtual interns last summer, undergraduates from around the world who worked together to build this pipeline. And I just want to acknowledge them here. Really, really remarkable group of young people with very little background in this kind of chemistry who figured it out very quickly and worked together really nicely to make this. So I'll give you just an example. I know we're getting a little tight on time. We decided to vet this initial thing on a really well-characterized reaction for which we had experimental data, and it is the caramelization reaction. You're probably all familiar with this. On the crust you get on flan or creme brulee is essentially a pure crystalline sugar that is then, when it is melted, undergoes a very complex set of reactions to give a really complex suite of chemicals. This kind of caramel here, right? This is table sugar, this is caramel, that has this very interesting flavor and odor profile, right? It can be nutty, it's sweet, it's burnt, it can be buttery. So you've made this very complex suite of compounds starting from a single molecule. And Typically, flan and creme brulee are done with sucrose, but sucrose is itself a time loop from glucose. We export glucose as a starting material. It's not particularly relevant to back up one step and use sucrose. The vetting of this experiment, or of this model, experimentally was done using a high field Fourier transform ion cyclotron resonance instrument at the National High Magnetic Field Lab in Florida. So this is able to obtain really fine resolution mass spectra, so we can be fairly sure that the compounds we're detecting are have the elemental formulas that we think they do. Okay, so we run this reaction network. It expands, it makes more compounds, and we can then ask whether we're getting mass matches of the experimental data with this experimental caramel with the model. And in fact, we do get almost a 95% match of the known molecules that have been detected using various analytical techniques, including FTI-CRMS. This is nice. We now have a system where we can actually throw in other kinds of starting materials in the simulation, and we can compare them against experimental models of, say, foremost chemistry or HCN polymer, Miller-Urey type reactions. And that's actually quite a fast process now that it's automated, right? This just matters maybe a couple of days to compute one of these networks and do the similarity comparison. So for visualization, this is what the caramelization of glucose network looks like. We're coloring the compounds as nodes according to the generation by which they appear. And you can appreciate that you start out with a single compound, and it gradually becomes more and more and more until it becomes this really just convoluted mess of things. But we can trace their etiology, right, by the processes which made them. And very quickly, at a touch of a button, we can compute what the physical properties should be of those compounds. For example, the C log P, which is a measure of water solubility, can be computed, at the drop of a hat for all of the compounds in this network. And now again, the potential utility of this is, can we say something about what compounds are actually present in the real world chemistry? And in fact, if we compare the number of possible isomeric graphs for CHO compounds over a certain mass range, we can see that we have this sort of exponential growth of the number of graphs as a function of the number of atoms or the mass. That outstrips very well the number of possible isomers that are known in chemical databases. So this black boxes here are the Beilstein database, and these triangles are the National Institute of Standards and Technology Electrospray ionization mass spectrometry database, the common library that analytical chemists use to identify unknown compounds. And then we have the components that are generated by the network. And the nice thing about this is, if you restrict yourself to trying to find compounds that are present in this network, by the time you get to 150, 160 AMU, you've actually compressed the search space by a factor of 1 million, right? So it allows you to now go in and say, well, I know that we have a compound of this formula. What can it possibly be? Well, you don't have to worry about the 180,000 possible isomers. You can worry about a much smaller number of things. Right. As I said, the nice thing about this is you can now go in also and trace loops around this network and say, are there pathways through this reaction space that are formally autocatalytic by any algorithmic definition you can come up with? And we can find many, many, many types of autocatalytic cycles within these sorts of spaces, and we can evaluate them with respect to their thermodynamic favorability. So I'd bring this home. I think this kind of graphical representation of chemistry is very useful for understanding the kinds of, chemical complexity that can be present in astrobiologically relevant reactions. you can construct a very logical and experimentally vettable workflow for interpreting these things. This can allow the discovery of entirely new types of chemistries and potentially models for the origin of life. And even when it fails, it allows you a way to refine your model and make it more predictive. So I'll stop there. I just want to thank my various collaborators who have contributed different aspects of the work over the years. And I'll thank you for having me, and I'll take any questions you might have.
Thanks, Jim, and a virtual round of applause. So a very nice lecture, detailing ways of how to come to grips with complexity, and I think that's bugging most people in the origins field who've thought about it for more than 10 seconds. Your head starts to spin. So who can I give the floor for questions, or whose question can I read out if you type them in the chat? I don't see everyone on the screen, so if you have your hand raised, so let me start off then. You say, okay, if we know rules in chemistry based on a century or more of doing organic chemistry in the lab, and of course that's right, but that chemistry is done always with a pretty restricted number of compounds in the flask, say. Are the rules going to be any, I mean, are we going to discover new things if we were to be able to analyze what happens if you have way more molecules?
So if I understand your question, you're sort of asking, are there new rules we don't know about?
Exactly.
That right? Yeah, that's always the possibility. And one of the nice things about this, and this is a future add-on we'd like to build in, is your model can overpredict or it can underpredict, right? If it overpredicts, you're including rules that you shouldn't be, right? And if you're underpredicting, there's a rule missing, right? So it's useful in that sense that it does allow you to say, wait, there must be a reaction here that we're not intuiting, right? And to try to figure out what that is, right?
I'm just wondering whether you think it likely that there are such things happening when the mixtures get more complex, that suddenly there are multi-component reactions that just simply haven't been on the radar because nobody thought about combining these precise reagents.
Well, that's a good question, and it leads to another question of what is a reaction, right? So so you can think of what we call elementary reactions, which are really single steps or single arrow pushing sort of processes. You have that at the one end of the spectrum of what is a reaction, and then you have things like the Bucher or Berg synthesis, which you can put a name on, but is actually 10 steps crammed into one, right? And so the question is, at this level of simulation, which is the better way to encode things, right? So if you do it at the elementary level, you can be pretty sure you're getting everything, but at the expense of computing a lot of things, that might be sort of concerted reactions, right? Does that?
Yeah. Makes sense. So there's a question by Martina.
Yes, I hope you can hear me. Thank you so much. It was a great talk, very interesting. My question would be, do you think it will be possible to also account for environmental conditions? Just, for example, simple things to begin with, and not that simple, I guess, but if you have aqueous surroundings or non-aqueous, because then also your reactions would be different depending on what the conditions are, right? And then it would lead to a different outcome of a molecule set. Do you think that will be ever possible, or is that too complicated?
I think it will, and I appreciate that. Every time I talk about this, somebody says, well, "What about kinetics?" We just made a conductivity map. It is still a little complicated to predict the kinetics of organic reactions in water. I think the techniques are improving quickly, and that would be a next step, is to try to build in some kind of model. That would be, I think, a computationally more intensive problem, and I'm not sure how accurate it would be at this step. There was another group based in South Korea that did something similar by just scraping the existing chemical literature and trying to say, well, what was the pH of this reaction? What was the pH of this reaction? And so kind of coarse binning mechanisms according to pH, right? That's not really a kinetic thing, but it gives you some variability according to environment, right? Whereas we called this, just say these are reactions that occur under slightly basic conditions, right? And we left out the acidic ones. So it's a good question.
Thank you.
Thank you.
So other questions? While you're thinking, then maybe I ask another one. So you're saying that when you do your, I think that was for the case where you looked at the caramel reaction, that's 95% of the product that you expect from your model, you also find in your mass spectrometer. But looking at it the other way, from the other direction, how many of the peaks you find in your mass spectrometer you can now account for by this, of course, limited subset of reactivity space that you've analyzed?
Oh, yeah, that's a good question. So there's two trains that are running into each other. Right? One is that FDIC-RMS is very good for doing the about 150 to 1,000, 2,000 mass range, right? Computationally, because you do have this kind of exponential growth of the network, you have to cap the network at some mass cutoff, depending on your computational resources, right? So we have some kind of finite overlap of the mass range of the model and the mass range of the measurement, right? Luckily, the lower mass range is a much sparser isomer space, right? But that has been explored very heavily by people using GCMS and more mundane bench tools, right? So the answer is that high molecular weight stuff, the model doesn't even get into it. It's too big, right? Right. So yeah, go ahead.
But where you can see things,
yeah,
analytically, how many of the peaks that you see there are accounted for by your model, and how many are not?
Oh, in the overlapping range, about 95% of the peaks are accounted.
Right. So yeah, it's 95% of the peaks you see, they come out of your model, not the other way around. 95%,
and including
well, the other way around maybe as well. But okay, so then you really account for the majority of.
Yeah.
So that is that. Do you think? I'm personally, I'm surprised that there's not more reactions going on that are actually not in your model, because I guess your model, I don't know how confined that was to a certain set of reactions.
I mean, not to oversell the system, and we think about this, like, why is it so good? If you think over the range of 0 to 200 AMU, only compounds that contain carbon, hydrogen, and oxygen, there's a lot of finite kind of integer hops and masses that are allowed, right? That are losses of water or losses of hydrogen or losses of CH-OH group. So it's not that every conceivable 0.01 mass is possible in that range, right? There's a lot of binning because of the isomerism. Does that make sense?
Right. Okay. So you're saying that if you were to push this out into larger masses, then you would see more. You would expect more problems.
There'd be more finite gradations of the chemistry, right? Now, I'm not sure that that's the point is to necessarily be able to explain all 100,000 peaks and a meteorite. The point is to be able to understand the reaction trends that generate the diversity, right, at the lower end of the mass range.
Yeah, I mean the big question is I mean, we always, and your point was also in the beginning of the talk, like you have this combinatorial explosion, and it's, I mean, if that's the reality, you have so little of anything particular that it's very hard to see any life emerge from that. And I side with that, that argument. But the question then is that really the right, is that really what chemistry does? By looking at the forest of peaks you get in a mass spectrometer, it looks like it, but because the numbers are so large, we might well be already overwhelmed, even though it's still a very small fraction of what could have happened also. So in other words, is the combinatorial explosion as that you see experimentally as large as you would expect it to be if you're take a very say pessimistic view on this, or are we actually seeing far fewer compounds than one might have expected? Lights turned off.
Well, that's a great question, and I mean now you're getting into more interesting sort of, I don't want to say they're philosophical but methodological questions. Let's say that you do one of these experiments that generates a mess on your bench, and there actually is some autocatalytic process that is evolvable and it's growing, and you shoot the stuff into your mass spectrometer and it's just a forest of peaks, and you're like, well, there's no adenine in there, so I guess this experiment was a failure. The point is, I think we may not even know what it is we're looking for, and we don't have enough of a comprehension of the chemical space to even be able to detect something that might be going on down in the weeds, right? And to be able to interpret it, because we can't a priori describe what the process would be, right?
Well, you just mentioned the probable autocatalytic system. I think that's a good one to look for. I don't know what the signs of it would be, but
well, then and what could it possibly be made out of, right? It has to obey some sort of laws of physics and chemistry, right? So
yeah, but will you stick it here? Yeah, I don't want to monopolize the discussion, by the way. I'm just looking at whether there are other people who have questions. I don't see any. If I overlook anyone, then please shout. Nobody shouts, so then maybe I'll continue monopolizing for a little longer. So this idea of an evolving autocatalytic system, I think that would be pretty cool if you found it. But how could you detect it? Do you have an idea about that? So what would be the signature of an evolving autocatalytic system without any preconceived idea what the molecules in it might be?
Well, so I think this is just me extemporizing here, but I think a real limitation to the way we've been doing a lot of this investigation in these areas is time. So you think of mass spectrum as a static thing, right? This is a measurement, the reaction is over, right? Well, what happens if you come back to the same reaction a year later? And can you curate your samples well enough that they can be compared across those kind of longer time points, right? How fast does something need to be for us to detect it, right?
Yeah, and you're not talking about closed systems in a sense, right? You put your compounds in, you put the stopper on your flask, and then off it goes, and then you might then keep monitoring over time, but then that will only go thermodynamically downhill. So
that's right.
You would, returning to the Urey-Miller setup, that was fueled, at least energy and input was continuous. So you would have, my hunch would be to run this in a way where you continuously supply, say, nutrients. So you run it in a CSTR as opposed to a closed system. Keep adding your food stocks, let it run, and then monitor, and then look for step changes in composition. I don't know if anybody's done that. That's apart from maybe the Yuri Miller, but even that wasn't really, I think, kinetically monitored.
So someone who specializes in oscillatory reaction dynamics told me that oscillating reactions are actually quite common and easy to find if you use flow reactors, right? Because you can vary the parameter space, and there's always some balance, or frequently some balance of input and output that will create an oscillator. Right? That's something interesting I heard. I think that's something to keep in your mind.
You talk to people that do heterogeneous catalysis. They actually, I mean, for them it was like they were never so interested. They wanted catalysis. They were not interested in oscillations, but apparently they're quite widespread in that field. And a nuisance because they basically, if you want to design a reactor and they start to oscillate on you, it's not good for your factory.
That's not what you want. That's right. Well, I mean, I also think, I mean, it's an interesting question. There was a paper came out in one of the big journals a couple of months ago. I don't remember what it was. Maybe PNAS, but they had studied the really long-term population dynamics of bacteria in sealed tubes, right? So they eat everything in the medium, they go into death phase, and then they still end up going into these population dynamics, where the survivors cannibalize each other, and that becomes a new species that can, a new variant that can live off the detritus of its dead comrades. And this goes on for years and years and years, right? Those little boom and bust cycles. So in the sense of like, even bacteria don't exhaust all of the free energy in a closed system over many, many, many years, right? So I don't know if that's something we have to worry about. A Miller-Urey experiment, for example, has so much chemical potential energy left at the end of the experiment. It's just a question of nothing has found a way to tap it, right? Does that make sense?
Yeah, but I guess life has of course evolved to tap just about anything.
Right.
But early life might have had a harder time.
Yeah, so I guess it becomes a question of what's your expectation of how frequent those kind of things are. And I think we're completely in the dark. Yeah,
exactly.
Yeah.
Okay, I'm looking for around again, but I don't see any further questions. Now I don't know, Bob, are you still around? Because the original schedule was we closed the YouTube thing at 5, and then then basically means you can have a somewhat more private discussion without the rest of the world watching with the people who signed up for this. Can I propose we switch to that now, since we I think have dealt with all the other questions?